Factors that regulate quantity of food intake. Regulation of the quantity of food intake can be divided into short-term regulation, which is concerned primarily with preventing overeating at each meal, and long-term regulation, which is concerned primarily with maintenance of normal quantities of energy stored in the body. Short-term regulation of food intake. When a person is driven by hunger to eat voraciously and rapidly, what turns off the desire to eat when he or she has eaten enough? There has not been enough time for change in the body's energy stores to occur, or it takes hours for enough nutritional factors to be absorbed into the blood to cause necessary inhibition of eating. Yet, it is important that he or she eat enough of food that approximate nutritional needs. Several types of rapid feedback signals are important for these purposes. Gastrointestinal filling inhibits feeding. When the gastrointestinal tract becomes distended, especially the stomach and the duodenum, stretch inhibitory signals are transmitted mainly by the way of vagi to suppress the feeding center, thereby reducing the desire for food. Gastrointestinal hormonal factors suppress feeding. Cholecystokinin, which is released primarily in response to fat and proteins entering the duodenum, enters the blood and acts as a hormone to influence several gastrointestinal functions such as gallbladder contraction, gastric emptying, gut motility, and gastric acid secretion. However, cholecystokinin also activates receptors on local sensory nerves in the duodenum, sending messages to the brain via the vagus nerve that contribute to satiation and meal cessation. The effect of cholecystokinin is short-lived, and chronic administration of cholecystokinin by itself has no major effect on body weight. Therefore, cholecystokinin functions mainly to prevent overeating during meals, but not play a major role in frequency of meals or the total energy consumed. Peptide YY. Peptide YY is secreted from entire gastrointestinal tract, but especially from the ileum and colon. Food intake stimulates release of the PYY, with blood concentration rising to peak levels 1 to 2 hours after ingestion of a meal. These peak levels of PYY are influenced by the amount and the composition of the food, with higher levels of PYY observed after meal with high fat content. Although the importance of this gastrointestinal hormone in regulating appetite in human is still unclear. For reasons that are not entirely understood, the presence of food in the intestines stimulates them to secrete glucagon-like peptide that enhances glucagon-dependent insulin production and secretion from the pancreas. Glucagon-like peptide and insulin both tend to suppress the appetite. Thus, eating a meal stimulates the release of several gastrointestinal hormones that may induce satiety and reduces further intake of food. Ghrelin, a gastrointestinal hormone that increases feeding. Ghrelin is a hormone released mainly by the oxyntic cells of the stomach, also secreted in less extent by intestine. Blood levels of the ghrelin rises during fasting, peak just before the eating, and then falls rapidly with a meal, suggesting a possible role in stimulating feeding. Although administration of ghrelin increases food intake in animal, however, its physiological role in human is still unclear. It is postulated that various oral factors related to feeding, such as the chewing, salivation, swallowing, and tasting, meter the food as it passes through the mouth, and after a certain amount has passed, the hypothalamic feeding centers becomes inhibited. The inhibition caused by this metering mechanism is considerably less intense, and it is of short duration, usually lasting for only 20 to 40 minutes. When an animal with esophageal fistula is fed large quantities of food, even the food is not going into the intestinal tract and it is immediately lost to the exterior, the degree of the hunger is decreased after a reasonable quantity of food has been passed through the mouth. Intermediate and long-term regulation of food intake. The feeding control mechanism of the body is geared to the nutritional status of the body. Effect of blood concentration of glucose, amino acid and lipid on the hunger and the feeding. A decrease in the blood glucose concentration causes hunger, which has led to so-called glucostatic theory of hunger and feeding regulation. Similar studies have demonstrated the same effect for the amino acid and the lipids such as the keto acid and for some fatty acids. That is, when the availability of any of these three major types of the food decreases, the desire for feeding is increased, eventually returning the blood metabolite concentrations back towards normal. Some neurophysiological study of function in specific areas of brain also support the glucostatic, aminostatic and the lipostatic theories. A rise in the blood glucose level increases the rate of firing of glucoreceptor neurons in the satiety center in the ventromedial and paraventricular nuclei of the hypothalamus. And the same increases the blood glucose level simultaneously decreases the firing of glucose-sensitive neurons in the hunger center of the lateral hypothalamus. In addition, some amino acid and the lipid substances affect the rates of firing of these same neurons or the other closely associated neurons. When there is increased glucose, amino acid or the lipid substances concentration in the blood, that increases the firing of the neurons at the satiety center and also decreases the firing at the hunger center. Temperature regulation and food intake. When an animal is exposed to cold, it tends to increase feeding. When it exposed to heat, it tends to decrease its caloric intake. This phenomenon is caused by interaction within the hypothalamus between the temperature regulating system and the food intake regulating system. This is important because increased food intake in a cold animal increases the metabolic rate and provides increased fat for insulation, and both of which tends to correct the cold state. Feedback signals from the adipose tissue regulate food intake. Most of the stored energy in the body consists of fat, the amount of which can vary considerably in different persons. What regulates this energy reserve, and why is there so much variability among individuals? Studies have shown that hypothalamus senses energy storage through the action of leptin. Leptin is a peptide hormone released from the adipocyte. When the amount of adipose tissue increases, that means that signaling excess energy storage, the adipocytes produces increased amount of leptin, which is released into the blood. Leptin then circulates into the brain, where it moves across the blood-brain barrier by facilitated diffusion and occupies leptin receptors at multiple sites in the hypothalamus, especially at the neurons of arcuate nuclei and the neurons of paraventricular nuclei. Stimulation of leptin receptors in this hypothalamic nuclei initiates multiple actions that decreases fat storage. It decreases production of appetite stimulators in the hypothalamus, such as neuropeptide Y. It causes activation of melanocortin receptors. It increases production of substances in the hypothalamus, such as corticotropin releasing hormone that decreases food intake. Increased sympathetic nerve activity, which increases metabolic rate and the energy expenditure. Decreased insulin secretions by the pancreas beta cells, which decreases energy storage. Thus, the leptin is an important means by which the adipose tissue signals the brain that enough energy has been stored and that intake of food is no longer necessary. Some physiologists believe that the obesity may be associated with leptin resistance. Leptin receptors or post-receptor signaling pathways normally activated by leptin may be resistant to activation by leptin in obese people. Another explanation for the failure of leptin to prevent increased adiposity in obese individuals is that there are many redundant systems that control feeding behavior, as well as social and cultural factors that can cause continued excess food intake even in the presence of high level of leptin. What are the importance of having both short and long-term regulation of feeding? The long-term regulatory system for feeding, which includes all the nutritional energy feedback mechanism, helps maintain constant stores of nutrients in the tissues, preventing them from becoming too low or too high. The short-term regulatory stimuli serve two other purposes. First, they tend to make the person eat smaller quantities at each eating session. thus allowing food to pass through the gastrointestinal tract at a steadier pace so that its digestive and absorptive mechanisms can work at optimal rates rather than becoming periodically overburdened. Secondly, they help prevent the person from eating amounts at each meal that would be too much for the metabolic storage system once all the food has been absorbed.